Thank you. Thank you. Thank you. Hello, everyone. Good evening. Really wonderful to have you here, and welcome. I have to give you a warning before we start. So I have this accent, and when I speak to a new audience, I may get a little nervous and then speak really, really fast, and then sometimes it's hard to follow what I say. This kind of was brought home when MGH, Mass General, when I was in training, decided to put together a video showcasing the diversity of the MGH workforce. And so they invited a number of people who came from different backgrounds, some funny last names, and come and tell what it was like to work at a place like Mass General. So there was a Mexican surgeon, there was a Haitian woman who worked in the kitchen, a Salvadoran custodian, a Nigerian chaplain, and myself, a trainee who grew up in Spain. And so we all come in, and then they're going to ask us what it's like to work at MGH, and I was very excited to be on this video that is going to be kind of the poster child for Mass General. My mom will love it, you know. And so they ask the Mexican surgeon what he thinks, and he says his things, and the Nigerian chaplain, and we all say what we think, and then they ask me, and I say what I think. 6 months later, they bring us back to watch the video that is now edited and ready for dissemination. And so we are all very excited, and there is the Mexican surgeon saying his piece, and the Haitian woman from the kitchen saying her piece, and the Salvadoran custodian speaking, and the Nigerian chaplain. And finally, I show up, and I start speaking, and I had to use subtitles because no one could understand. So this is not a subtitled talk, so I will try to be slow and so that you can follow. Okay, let's start by getting everybody on the same page. What really is diabetes? And here's an image of a glucometer. This is a device that every person with diabetes needs to use multiple times a day, pricking their finger to check their blood sugar and make sure that it is in the normal range. Now, you will note that most ads on TV or in magazines with glucometers have a perfectly normal blood sugar. It's amazing how these devices make you have a normal blood sugar. Because diabetes is really defined by the final common pathway of what is your blood sugar. At the time after 8 hours of a fast, if it's higher than 126, or randomly, if it's higher than 200 and you have symptoms of high blood sugar, like frequent urination or increased thirst, or any blood sugar higher than 200 after a standardized 2-hour glucose tolerance test, when you take a glucose drink and we measure your sugar, or an average measure of your sugar in blood caused by how sugar molecules, glucose molecules, get stuck on hemoglobin, which is the protein that is carried by your red blood cells, and that is higher than 6.5%. All of that gives you the definition of diabetes, a high blood sugar in blood. It needs to be repeated twice to make sure that this is a permanent condition. And really, in essence, it probably is many different things that lead to that final common pathway of high blood sugar, but they all result in similar complications. So the definition is based on a final output of many different molecular, biological, cellular pathways that lead to this derangement. So it really is all about sugar. So here is this shop of sugar. Now, where did I take this picture? I took this picture in the connector that connected the hotel to the American Diabetes Association national meeting. So it kind of goes to show you how difficult it is to really make inroads in diabetes. You know, the other thing that this reminds me of, so I have kids, and in our school, sometimes they ask parents to come in and tell the children about our work. And so my wife is an ophthalmologist, and so she gets invited to speak. It's very easy for her to say what she does. She works on diseases of the eye, and kids, first graders, understand what the eye is and why you need to see and why a doctor can actually help your vision. Very difficult for me to tell people that sugar is bad, you know, to tell first graders to come in that sugar is bad and explain what does a doctor do who deals with blood sugar. So I was commiserating about this with another parent of one of my daughter's classmates, and she goes, well, you think you have it hard. I'm a fertility doctor, so you tell me what I tell them. Now, diabetes is classified typically in type 1 and type 2. Type 1 is present in about 10% of the people with diabetes, and type 2 is the most common form in about 90% of people. And the main difference has to do with type 1. The process that leads to high blood sugar begins with your immune system attacking your own beta cells, which are the cells in the pancreas that make insulin, which is the hormone that controls your blood sugar. So those beta cells, because of some deranged process in the immune system, get killed by the T cell response, and then you don't make any insulin, and therefore you cannot drive glucose that you consume or that you make in the liver into muscle, fat, or other tissues, and that leads to high blood glucose. This is a condition of absolute insulin deficiency, and the only way these people can survive is by taking insulin to be able to replace the insulin they no longer make. Type 2 diabetes, the very common form of diabetes, is caused by a different process, kind of ill-described. Together, obesity, genes, and other factors contribute to make the target tissues where insulin is supposed to work be less responsive to the action of insulin. And because they are less responsive, then they don't take up glucose as efficiently as they should, and glucose rises. So the pancreas typically tries to overcome this defect by making a lot of insulin. And in people who have a very well-functioning pancreas with lots of beta cells and good function, then they may be able to make enough insulin for a while to make sure that the glucose continues to be driven into tissues. But when that manufacturing and secretion of insulin is not sufficient to defend your glucose, then glucose rises, and then you become a diabetic, typically in adult age, where this happens typically in a young age. Now, the big problem with diabetes, of course, is not walking around with a sugar of 200 or 250. The big problem is that the extended exposure of your tissues to high blood sugar can lead to really, really serious complications that completely limit your quality of life and actually leads to increased mortality and morbidity. So blood vessels get affected by high blood sugar, and the large blood vessels that feed the brain or that feed the heart can actually be occluded by this process, and you get a stroke or a heart attack at a premature age. You can have small vessels, the vessels that have to do with bringing blood to the retina so that you can see, or that feed in the kidney. Those also get affected, and you can end up with blindness, or you can end up with end-stage kidney disease and on dialysis. The vessels that feed the nerves, or that go long lengths down your leg to irrigate your limbs, can also be affected, and then can lead to amputation. So there's a number of many very serious complications that happen in people with long-standing, ill-treated diabetes, whether it's type 1 or type 2. Again, the complications are common to this final common pathway. Now, clinicians like myself recognize that it's not just type 1 and type 2. There's other subtypes that one can try to define in the clinic that I won't go into here, so the pie gets a little more complex. Now, it turns out we've known about diabetes for a long time. So 1500 BC, there's a papyrus in Egypt that described patients who had increased urination. Most likely increased urination is caused by all the sugar in your blood. Your body tries to get rid of all that sugar, puts it into the urine, and so people who have increased urination sustained for a long time often have diabetes. And that's the first description, one of the very first hormone disorders to be described. Now, in endocrinology, we like to give the drug that the person needs to overcome the defect that the body has. And for diabetes, we've had the molecular therapy, the therapy that actually solves the problem, insulin, available for over a century, almost over a century. So Frederick Banting and Charles Best, shown in this picture, Banting and Best, working in Canada, were the first people to isolate insulin from the pancreas of dogs. So the investigator was Banting, who was working on other things and decided to maybe focus into this for a summer, and then asked for help from the chair of biochemistry in his department to give him someone who can help him. And then there were 2 students that were offered to him. And then he decided, this is a simple problem. I only need 1 student. And so the 2 had to flip a coin. And so that flipping of a coin was really the difference between a Nobel Prize and not a Nobel Prize. And so during that summer, they were able to isolate insulin from the pancreas of dogs that were first administered to patients a year later. And then a year later, Banting and MacLeod got the Nobel Prize, and Banting generously shared the proceedings with his student Charles Best. And then Eli Lilly, an American company, took over the extraction of insulin and was able to commercially make it available for the first time in 1923. Now, people who did not make any insulin, that have a high blood sugar, and the treatment that was available for those children was basically starvation. If the way you become sick is because your sugar is high, what I will do is I will limit the amount of sugar you consume. That's the only way I can do. So these kids were starved, and of course it was an irreversible process that eventually led to their demise. And so when insulin is finally introduced, it was really a miracle. And you had children like this who were in some of these sanatoria, basically starved and miserable, and then with the use of an injection from insulin isolated from pigs or from cows, which is where you could get it in large amounts, and produced by Eli Lilly, you could rescue at least their ability to lead a fairly normal life, except he's not that happy because he has to take injections every day to maintain his blood sugar normal. But at least you can take what's a universally fatal condition and transform it into a chronic condition. Now, it turns out that of all the proteins available for scientists to develop methods to sequence proteins and come up with a sequence that comes up with the building blocks of a protein, the one that was selected for the very first attempt was insulin. And so Frederick Sanger, in 1952, produced the first sequence of human insulin. And then that also led to the first of his 2 Nobel Prizes. So his first Nobel Prize was for protein sequencing, and then I guess he liked the sequencing part of things. So then he invented DNA sequencing, which eventually gave rise to the sequencing of the human genome, for which he obtained the 2nd Nobel Prize. So now having the sequence of human insulin, and then with the advent of recombinant DNA technology, where you can take DNA and then put it into bacteria and grow a human protein in bacteria, recombinant human insulin became available in 1978, finally commercialized in 1982. And now we have this proliferation of insulins with which people are treated. So the disease known for a long time, the molecular therapy that restores normal blood sugar available for at least a century, the human form, which is the very one molecule that you're missing, being able to be given to patients for at least 30 years. And yet, today, 415 million people worldwide have diabetes. There is incredible mortality. There's incredible morbidity in childhood and incredible healthcare expenses. So there's something that is just not working well for the care of people with diabetes. And so here is the panorama. And the first number by continent is what the diabetes prevalence is in 2015 in the latest data from the International Diabetes Federation. And then the projection for 2040 at each one of the continents. And you can see that these figures are staggering, and that no one really can withstand the burden on healthcare systems that this is going to bring, particularly in developing economies. How is India, how is China going to be able to cope with the onslaught of heart attack and stroke and dialysis and blindness that is upon them because of the very, very large numbers of diabetes that are beginning to flood their clinics? And when you are a practitioner, you see it up front. I have seen a young father of 2 children, 41, have his first heart attack at age 41. I have seen a person younger than 40, an immigrant, who first lost a toe and then another toe and then his foot and then eventually succumbed to overwhelming infection at age 37 in our hospital. And I have seen blindness, the curtain of blindness descend in middle age. So there's all these incredible morbidities that are really threatening not only the developed world, but increasingly the developing world, and threaten to cripple, really, the global economy. And why is that? So here's what's happening in the United States for obesity over the last 20 years. So this is a map where states are coded by the number of cases of obesity, defined as a body mass index higher than 30. And so you see in increasing shades of yellow and then orange and then red how obesity has grown in prevalence across our country in the last 20 years. You see Colorado is like the one spot where you may want to move to if you want to have a good weight. And of course, that is what drives, what fuels the epidemic of type 2 diabetes on its heels as well. Now, it's not that we don't have other treatments. So beyond insulin, we have 11 other drug classes that we can use to treat that high blood sugar. 11 drug classes. Drug companies have been busy and trying to come up with medications that lower low blood sugar. And this is a very complicated, you don't have to read this, by the way. This is a very complicated algorithm that is used recommended by the American Diabetes Association and the European Association for the Study of Diabetes for the treatment of diabetes worldwide. You see that metformin is the very first drug that we use, and then there's a number of different options that we can put in, and then if those don't work, then we add another one, and you can see all the different combinations that one can use to treat diabetes. Now, there's 3 problems with this approach. Number 1 is that these drugs just lower blood glucose, but they don't cure the disease. They have to take them forever. You don't necessarily reverse the process that led you to high blood glucose. You simply control the end result, which is the high glucose. The second problem is that this is a one-size-fits-all approach where everybody starts with metformin. And then if that doesn't work, trial and error, you do something else. And if that doesn't work, then you do something else. Not really tailored, individualized to the type of diabetes, the type of derangement or process that led that person to have diabetes in the first place. And then the third problem is that despite this armamentarium, people don't take it as they should. It doesn't really have the uptake that you would want in the population. So there really is an urgent need for a major change in how we approach the diagnosis, the thinking, the pathophysiology, and the therapy of type 2 diabetes. We really think that is much more heterogeneous than previously known, that there is many different ways in which one can reach hyperglycemia, and that the way you treat people should be tailored to the particular defect that they carry. And we don't necessarily treat glucose. Like cancer is not just growth. We don't simply treat growth and do something that limits growth. We have to go to the molecular cause for the cancer and then give a drug that addresses that defect. And so the same process needs to happen for diabetes. And so the good question in front of us, and what motivates our research program and many investigators worldwide, is can we use the plethora of information, of exposures that people have, of the genome that we now have at our fingertips with all the bases that are sequenced, what genes are active, what messages they make, what proteins they produce, what metabolites are the result of the action of those proteins. Can we gather all of that information and have a coherent picture that for each person or group of persons in front of us, we can come up with a molecular diagnosis that really defines the disease for that person, and then apply the therapy that corrects the pathway that is deranged for that person. And so the very first thing that we need to take stock of is the existence at our fingertips of the human genome. And I think here we need to take a pause for reverence for the moment. So all of us, I think, all of us were alive when the human genome was sequenced. And in 2003, the sequence is published, and it was a momentous event, not only in the history of science, but in the history of humankind. I think when it comes to politics and history, we kind of go in circles a lot. So we kind of elect the same kind of people, and we get into the same kinds of wars, and all the conflicts tend to be the same, kind of a circular process. But when it comes to science, it's all incremental. There's a before, and there's an after. And there's a discovery that transforms the way things are known, and there's no going back. There is no going back. And so, for example, there was a before and after. There was a world of pain and suffering when you had to have an amputation just by biting on something and drinking a lot of whiskey. And then ether comes along and you have anesthesia. And you can undergo surgery without a touch of pain. And my wife can deliver 4 children with an epidural anesthesia and not feel a thing, whereas pain and suffering had been the common stock of labor. And so at MGH, ether gets introduced. And then I already told you about how insulin transforms a uniformly fatal disease into a chronic disease. There is antibiotics, and people could die from a simple tooth infection or from an abscess, and now with antibiotics, we don't think twice about going to the pharmacy and getting our Z-Pak and getting off with our pneumonia. And more recently, we have seen people with HIV/AIDS who have been rescued also from a threat of an immediate demise and a horrible death with terrible complications, and now they're treated with medications that transform it into a chronic disease. So there's a before and an after, and I think for the human genome is the same thing. There's a before and an after. And what we are here to do is to try to see whether with the information that the genome has afforded us, we can transform how we think of medicine and how we treat patients, and particularly with type 2 diabetes. And so for type 2 diabetes, over the last decade, our group and many others working together, for the most part in international collaborations, have uncovered at least about 100 places across all the chromosomes in the genome, shown here as these little yellow dots, where a human variant, a variation in DNA sequence, increases your risk of developing type 2 diabetes, beginning to have the genetic architecture, the changes that lead to changes in proteins and then in pathways that might lead to hyperglycemia and thereby give you the disease. And so for the rest of the talk, what I'd like to tell you, what is this genetic data, this explosion of information that has only taken place in the last few years, what does it tell us about how we can treat diabetes? Can we identify special forms of diabetes that are very specific in a small group of people? Can we use the genetic information to predict, in general, in the population, who's going to get diabetes and who isn't? What about some maybe things that work in special populations? Or what about using it for drug discovery? Can tell us about targets that might be harnessed as a way to develop new therapies? Can we use it diagnostically? Can we use it to predict response to medications, response to side effects? And can we apply it for population health? And so I will use the rest of the talk to give you a little vignette for each one of these. So here is a special form of diabetes. It's called maturity onset diabetes of the young. This diabetes that occurs maybe in about 3% of people with diabetes and is inherited from father or mother to child in about a 50% ratio across generations. So what this shows is a beta cell that makes insulin. And the key function of this cell is to respond to glucose, sugar in blood, by making the insulin that you need to drive that glucose back into tissue so it can serve as the fuel that those tissues need. So glucose comes in, and then there's a number of transformations that take place. The very first guardian of the gate is glucokinase, GCK. It's an enzyme that adds a phosphate group to that glucose, and that glucose enters a cycle, and there's a number of channels, potassium channels, calcium channels, that work in concert to eventually drive insulin out of the cell in a very quick process within 5 or 10 minutes after the ingestion of glucose in adequate proportions to the amount of glucose ingested. And that measurement of the glucose that you take and how much insulin to be made is all driven by this guardian of the gate of glucokinase, the gatekeeper that tells you I will be as active as I need to be depending on how much glucose I'm seeing inside of this cell. Well, it turns out now there are mutations in this glucokinase where you can have disruption of at least one copy of the enzyme, and that disruption elevates the so-called glucostat, as opposed to a thermostat that regulates room temperature, glucostat that sets what the glucose level is supposed to be in the body, but not by much. So instead of living at 100 milligrams per deciliter, then you live at maybe 130 or 140. And it's simply an elevation that stays constant for the rest of your life, and the rest of the machinery is intact. So that type of maturity onset diabetes of the young, or MODY, type 2, is really not a very serious form of diabetes. It's simply living at a little bit of a higher glucose level, but then you don't develop complications. You really don't need therapy. You don't need to escalate therapy. You don't have progressive beta cell failure. So being able to make that diagnosis is crucial. And it was really brought home to me with my patient, Laura. So I can use her name, who comes to me, immigrant woman who is trying to get pregnant. And in the course of trying to get pregnant with some difficulties, they find that she has a high glucose level. She's very athletic. She eats very well. And she's horrified by this idea that at age 28, she has diabetes. And so she comes in. The picture fits the potential diagnosis of MODY, but you need a genetic test to be able to prove it. And so you do the genetic test, and now you can tell her, look, this is just a normal variant in a way. You will live at a little bit higher glucose level. From now on, you do have diabetes because you exceed that threshold that we use to give you the diagnosis, but you won't need medication. And it won't really affect your baby that much. We'll need to follow your pregnancy closely to make sure. And as long as you keep having a very healthy lifestyle, then you shouldn't have any problems with this. You won't progress to blindness and kidney disease and so on. And so she was extremely relieved. So right now in the clinic, we can use genetics to, at least when we suspect one of these special forms of diabetes, be able to make a diagnosis that provides great reassurance to our patients. Now what about prediction? So can we use the 100 or so places in the genome where we know the variation increases risk of type 2 diabetes, deploy it in the population and say you are at higher risk of developing diabetes and therefore I need to follow you more closely, I need to recommend more exercise, really pay attention to your diet and do things that will prevent that from happening. So in this experiment that I carried together with my colleague and friend James Miggs, who works for the Framingham Heart Study, we asked the question of whether a genetic test based on the genetic information known at the time improved the ability of a clinician to predict diabetes in a patient over common clinical factors that are available in the clinician's office. So when you come to my office, I can measure your sugar, I can measure your weight, I can measure your height, I can ask you about family history, I can look at your diet, I can look at your cholesterol. I can put all of that together and come up with a prediction model that tells me you have X chances of developing diabetes, right? And so the way the curves are shown, so a test that only gives you 50%, which is a completely useless test, it's like flipping a coin, you have a 50% chance, would fall under the diagonal on this curve. So the performance of the clinical test, you want a test that goes as close to the square as possible to be closer to 100%. And we have two curves here. You can't see two because they're perfectly superimposable. One that simply tests the simple clinical model that uses the values that I can achieve in my clinic, and then the other one that includes the genetics. And you will see that the simple clinical model gives you 90%, meaning that out of 10 patients, I can guess correctly in 9 of them based on clinical factors alone. And then when you add genetics to that, it is perfectly superimposable. It really doesn't add much. We are so good at predicting diabetes already clinically that these genetic factors don't simply add a whole lot. So you will not be getting genes done in your clinic when you first come in to improve on prediction. So James was very happy to publish this in the New England Journal of Medicine. And then only a few months later, I happened to go back to Spain and then stopped in Madrid and I went to the Prado Museum. And in the Prado Museum, there's Velázquez, one of the major painters from the Spanish Golden Age in the 1600s. And he has this painting called The Drunkards. And it's amazing, the resemblance. I mean, look at this. Unbelievable. Like, this is not Photoshop. This is like the real painting. And it's, he was like celebrating the publication of his paper with Bacchus, the god of wine, and his friends. So anyway. So given that all these different genetic variants don't really help much for prediction, the model that has been proposed is the palette model by my friend and colleague Mark McCarthy at Oxford, where he likens the genetic factors that contribute to diabetes as the different colors that are in a painter's palette, following on the painter's analogy. And so yes, there are parts of the palette that are clearly red, and maybe a single genetic variant, like my patient with MODY diabetes, I can tell you, you have this type of diabetes, or clearly blue. But for the most part, they tend to be a mix, like a mesh of many, many factors that interacting with the environment, with diet and exercise, all contribute to your risk of giving you diabetes. And so different people have a different constellation of factors, and so they could be where one factor really has a huge impact, but then others that are kind of more in the middle, and then of course people move across the palette through the course of their life. So it doesn't appear that we will be able to really use genetics, strictly speaking, for the entire population to predict or define, but maybe for some special groups. And so one such group is the Inuit. The Inuit who live in northern Canada and Alaska and Greenland. That's kind of the entire territory where they tend to live. It turns out that recent work has uncovered one genetic variant that increases risk of diabetes tenfold, and is actually quite common, but in this population alone. So it's not present in any other population, only in the Inuit, but it's about 17%, so that's not trivial. And it works specifically by really increasing the insulin resistance, suggesting that if you happen to be a clinician taking care of this population and living in this northern part of Canada, I always wonder, it's amazing that there's an entire other country north of Massachusetts, but it's true. Then you would be good to think about this potential cause for their diabetes, because you might treat them differently depending on the medications that happen to work with insulin resistance as opposed to the other medications. So we carried out a study in Mexico to see maybe in Mexico we can find something that is also different in people, and so we published this recently. And we did uncover one mutation in a gene called HNF1 alpha that is carried by 2% of Mexican patients with diabetes that increases risk of diabetes fivefold, so not as much as the Inuit, but also significantly. And it turns out that there's a different type of MODY called MODY3, where people who have other disruptive mutations in this gene respond better to a class of drugs called sulfonylureas. So the idea would be that if we identify the carriers and we prove that they also respond better to sulfonylureas, again, that would guide the type of therapy that we apply to these patients. And again, you may think that 2% is a small number, but really there is 7.3 million patients with diabetes in Mexico. That makes about 140,000 people who carry that variant as the cause for their diabetes, which is basically all these people into Campo de Azteca Stadium. So it's not a trivial number to treat, perhaps in a more targeted fashion. So we conducted another genetic study in Mexicans where we tried to find kind of the common variants that have smaller effects. We found a new gene, SLC16A11. It was a study of 9,000 people from Mexico. So it has an interesting history. So it turns out that this part of the genome that was altered to increase risk of type 2 diabetes came into modern humans via Neanderthals. Now let's think about this. So when I went to high school, I learned about human evolution in a very linear fashion. So first was Australopithecus, and then was Pithecanthropus, and then was Neanderthal, and then Cro-Magnon, Homo sapiens. It was all like linear and one after the other after the other. Well, it turns out that's not how things happened. So Neanderthals, who share a common ancestor with modern humans, coexisted with modern humans in northwestern Europe. So they lived at the same time. They came from a different part of the tree, and they fought for space and for resources together, and there was mostly not a very friendly relationship. But there was also some instances of what scientists call gene flow between the two populations. Hanky-panky for the popular press, and they mixed. And so some of those genes came over, and it turns out that this portion of the genome that increases type 2 diabetes in Mexicans came into modern humans at that time. And they migrated eastward and rose in frequency. The further east you go across the Eurasian landmass, the higher the frequency. It was about 2% in Europe, about 11% in Asia. And then it crossed the Bering Straits into the Americas and rose to about 40 to 50% prominence in the Americas. It gives you a higher risk of diabetes by 28%. Here's the difference in its prevalence. And so that difference explains to about 20% of the difference that you see in increased risk of diabetes in people of Latino descent is caused by this single variant. Now, this is interesting from the population health, but then can we use this new pathway, this new gene, maybe to develop therapeutics? And so in a paper that we just published, we were able to demonstrate that this gene encodes for a protein that transports a metabolite across membranes of the cell in the liver, pyruvate and other fuel metabolites. And in the normal situation, you make the protein finds a chaperone called BSG that carries it to the surface of the cell, where it can serve as a transporter that brings these metabolites across. Now, if you happen to carry the variants that came into us from Neanderthals, it turns out that you have two problems. One, you don't make enough of the protein. And second, the protein that you make doesn't really bind very well to the chaperone. So you end up with very, very few levels of the protein in the membrane. You can't really do your work. That leads to many changes that we see in people with diabetes today. And so that illustrates the pathway for what a drug company might want to do. The key thing is we need to find ways in which we can get this protein to the membrane in appropriate amounts so that it can do its work, just like you do, for example, in cystic fibrosis. So this is now the next stage. This paper was just published, and this is what we're trying to work on. So what about the use of genetic information to make therapeutic choices and decide which drugs are going to work for the person in front of me? So you have when you're in clinic, you have a patient population that comes from many different walks of life. You have a number of drugs. In diabetes, we have 12 different classes, each one with multiple drugs within each class. And the question is, well, some people you will give the drug. They will have some side effects. It will be toxic for them, but it will also help them. There are some people for whom the drug will be like a placebo, does nothing at all. There are some people where you will hurt, but actually not help at all. And you want, of course, to find the people for whom the drug is not toxic, but is beneficial. And so I really don't know. When I have 12 different drug classes and somebody walks through my door, I'm told by my professional organizations to start with metformin for all, and then if that doesn't work, it's kind of trial and error. And then keep trying until you find things that work. Doesn't seem like a very efficient or rational way to go about therapeutic decisions. So can we use pharmacogenetics? Can we use genetic information to make decisions that are a little more helpful for people? So there is precedent. There is precedent in something called neonatal diabetes. You've seen the beta cell before. Now it turns out there are mutations in this channel, this potassium channel, that give diabetes in the infant stage. So from newborn to about 6 months, 1 year, this diabetes manifests itself. And you know what it's like. You know, you have your child, and you're so excited, and then maybe you're a new parent, and you're trying to learn how you swaddle. I can never swaddle. You can use the diapers, and then all of a sudden, 2 or 3 months into this, the child becomes a little lethargic and irritable, and the diapers are heavy because they're full of a lot of urine, and the child seems to be very thirsty. And so you take him to the doctor, and the sugar is 300, and so the pediatrician tells you your child has diabetes. And so now all of a sudden, you're thinking that I'm going to have to prick this child multiple times a day to measure their blood sugar, and I'm going to need to give them multiple daily insulin injections. Prior to the discovery of these mutations that give you neonatal diabetes, this was thought to be type 1 diabetes, the autoimmune destruction of beta cells that completely destroy your beta cell complement, so that you need to give insulin for the rest of your life. And you can see how the life of that child through infancy and through toddlerhood and adolescence and then young adulthood is completely changed forever because of this. Now, it turns out that there are these mutations that give you neonatal diabetes that simply disrupt the way for this system to transduce or transfer the information of glucose coming into the cell and insulin secretion. There is a block here that breaks the entire chain of events. So there's a clinical observation that didn't quite jive with the idea that this would be type 1 diabetes. For type 1 diabetes to ensue, you need the destruction of every beta cell in the body. That takes time. That can't happen at 3 months. That takes a year, 2 years. And so really odd that these children would show up to the emergency room with diabetes diagnosed within 3 months of birth. And so the gene gets found, and the idea that you might be able to treat this differently came from this observation, true story, of this Brazilian man who was born in rural Brazil in a family of poor means and was diagnosed with diabetes at a young age and had to take insulin for many years, and then eventually decided that he was tired of insulin and that he couldn't put up with the glucometer and the injections and the syringes and decided to stop altogether. Now, you've seen what happened to children who don't take insulin who have type 1 diabetes. They don't survive. He did. He survived. He went on to law school. He got a degree. He became wealthy. He got married, had children, and now his 2 children also had diabetes at a young age. Now, he's a man of means, so he decides, I'm going to consult with my endocrinologist and go to the world's expert to tell me what I have, because this must be genetic, because my children have it and I have it. And so he contacts Dr. Andrew Hattersley in Exeter, England, who is the world's expert on genetic forms of diabetes. And then the clinical observation comes from saying, you cannot have type 1 diabetes because you would have died. You have something else. And if what you have is a genetic defect here that blocks the ability of your beta cell to transfer the glucose information into insulin, all we need to do is to simply overcome that defect with a drug, because the machinery is all in place. The cell is not dead. The cell is there, ready to make insulin. And so it turns out that this potassium channel is the target for the sulfonylurea class of drugs that was the first class of drugs, oral drugs, developed in the 1940s to treat diabetes. And so with this insight comes a study where you now take people who have mutation in this potassium channel, and then you measure how much insulin they're going to make. You give them glucose, and of course they don't respond to glucose. They never have. That's why they have diabetes. But you give them tolbutamide, one of these sulfonylurea drugs, and they have a robust response to glucose. These cells were just waiting to be awakened, and they did. with a drug. And so all of a sudden, you can take this diabetes that was treated with insulin injections multiple times a day and finger sticks, and with a single pill once a day, you can treat their glucose. And you completely transform their lives. One of the first American children that underwent was Lily Jaffe, and so there's a lot of story. It was done at the University of Chicago, which is now kind of the center of excellence in the United States, where this type of diabetes is taken care of, and you can see how happy she is now with her glucose is showing. It's a little blurry, but it's a perfectly normal blood sugar, like most glucometers will show you. It's a perfectly normal blood sugar. And then she's grown up to be a young girl. There's you can Google Journey to a Miracle, the video that kind of shows her life story. So for the minority of patients who have this very rare condition, it really is transformative. And so the question that we're really very actively working on is, can we use the same type of genetic information to guide how a physician or a nurse practitioner, a general practitioner, a primary care doctor can be in a clinic and then decide that I'm going to give you the drug that will work for you. Now, it can also be helped diagnostically. So sometimes it's difficult to decide whether the patient in front of you has type 1 or type 2 diabetes. That has radical different implications for how you treat it. And so when the person comes in, maybe atypical because they don't really fit the textbook definition. And so you could have an older person who has new-onset diabetes and not quite typical of type 1, but is lean, but there's no antibodies, so you don't know, is this really type 1 or type 2? We have a younger person, so should have type 1, but this person is obese because obesity is growing and growing, so to speak. And then this person has antibodies they give you, so you don't really know. It turns out that you can now use genetics to try to distinguish what type. And in those difficult cases for the clinician, you can also make a difference. So what about for prevention and public health? So it was mentioned in the introduction that I work closely with the Diabetes Prevention Program. This was a clinical trial conducted across 27 centers in the United States to see whether we could have interventions that stopped, delayed, or prevented the onset of diabetes. So there was 3,200 participants, all with impaired glucose tolerance, so pre-diabetes, a high risk of developing diabetes. They received either placebo, which was a standard of care at the time, really nothing active, a very intense lifestyle modification with diet and exercise, a personal trainer, an exercise program, and a nutritionist assigned to them. And then they were followed for 4 years. DNA was collected, and the onset of diabetes was measured here. And you can see that both metformin and the lifestyle intervention were quite effective at delaying the onset of diabetes, with a 58% risk reduction offered by the lifestyle intervention and 31% by metformin. So the question is, okay, we have interventions now that work at preventing or at least delaying the onset of diabetes. Can we use genetics to really ask questions about what people should be put into these kinds of interventions? So the very first gene we tested was TCF7L2, which was one of the highest diabetes genes that were known at the time. And for people who carry 2 copies of the gene, shown here, you see that the incidence of diabetes about doubles that of the other 2 types of genetic groups. So if you have 2 copies of the gene, you have twice the risk of diabetes that no copy of the gene variant or a single copy of the gene. If you are in the placebo arm, meaning you get no intervention. Now, if you get the intensive lifestyle intervention, regardless of your genetics, you have the very same risk as everybody else. This is an incredibly positive message. That means that if you get off the couch and you engage into a fairly moderate amount of exercise, about 30 minutes of brisk walking 5 days a week, and then a reasonable diet, you can overcome the bad deck of cards that was dealt to you through inheritance by this particular gene and have the very same risk as everybody else. You can do something about your genetic risk. You're not doomed to develop diabetes if you can do something about this. That's for the one gene. So Marie-France in the group was interested, what about if we look at all the genes we knew at the time? And so developed a genetic risk score where genetic burden is now divided by groups in 4 equal groups. And you see that when you are in the placebo group, meaning the people who don't get any intervention, your genetic risk travels with your incidence of diabetes, and the higher the genetic risk, then the higher the risk of diabetes. But if you're in the lifestyle intervention, again, that risk is completely flat. You have no increased risk. So in this case, nurture, the environment, can overcome nature or the genes, which stands to reason. The same way that the bad environment has given the epidemic of obesity and diabetes that we see today, if we can do something about that environment at the individual level, we can also reverse the risk back to baseline. So as a result of all this, our former president, President Obama, introduced first in the State of the Union and in a special ceremony in the White House, is the DNA double helix. I think it's the first time it went into the White House. introduced this precision medicine initiative, a very ambitious project in which 1 million Americans will enroll in the All of Us cohort. It's called All of Us. And then information will be gathered, genetic information and dietary information and behavioral information. All that will be gathered for many diseases across many different states to be able to really guide the next generation of therapies that really have to do with precision, with giving you what you need at the time you need and only what you need without side effects. And so the question, is this possible? Can we do this? And it's very ambitious. So I will go back to another president who in 1961 gave this speech at Rice University in Houston and then had this incredible challenge of getting us to the moon. Now, in 1961, no one had any idea, first of all, how to get to the moon. You know, you've seen, I mean, it was like a contract with like rubber bands, you know, and glue. I mean, it was, and tin. That's what they had to contend with. And why? No one had any clue that getting to the moon would improve life on this planet. But we could develop the technology. We had the ability to do it, and so we will do it. We will go to the moon and figure out whether going there helps and whether life here is better as a result of all the technological advancements that are taking place as a result, and then the pride and the confidence that arises from being able to triumph in such an ambitious endeavor. And so the same way happens here. I think the Precision Medicine Initiative, the idea that we can be able to understand the molecular makeup of diabetes one day and use drugs that work in a person at the individual level, that's a very ambitious, tall order, but something that can be achieved not in a century, not in two, in a professional lifetime. And that's what we work here at the Broad. Now, I've mentioned two inspiring initiatives from two former American presidents. Now, they both happen to be Democrat. And this is, I know that we are in Massachusetts, and this is Cambridge, but you know, it's an apolitical talk. So I think we need to give some balance. And many of you are thinking, oh, what's he going to do now? And so I think I want to also bring in a former president who also did something that inspired me, and it's the idea of tearing down walls. And so I think we're all also alive in 1987 when Ronald Reagan goes to Berlin and tells Mr. Gorbachev, tear down that wall. Of course, he was very much already in the process of doing so. And that wall comes down. And so when I think about diabetes and the problems that face us, you know, I think about the walls that prevent us from reaching discovery on the other side of that wall that we need to get over so that we can break down the barrier that keeps us away from discovery. The walls that imprison people into a disease from which they cannot escape. And right now, we don't have anything to offer them for cure. We only have things to offer them to keep going. And so we need to break down those walls that make them go free. And then the walls that divide us, the walls that divide researchers, the walls that divide institutions, organizations for this common mission of being able to finally rid the world of diabetes. There is a clear example here where we stand today at the Broad Institute, where Harvard and MIT, which lived in the same city but had not come together for centuries, I guess, create the Broad Institute as an example of where walls can be broken and then changes can be made. And now Broad and MGH working together to try to solve diabetes, and so you can empower people to then take control of their lives, working together for that common goal. So of course we work as a very, very big team. We have three groups of very talented people of many different walks of life and levels of expertise, types of expertise that work to make this happen. We collaborate with people across the world. I have fantastic mentors, many different consortia that we work with, many people who are kind enough to fund us, and then, of course, the home team that allows me to take an evening to come and talk to you and tell you about diabetes. So with that, thank you so much, and we're happy to answer questions.
Thank you very much. And fine, I will start going to the gym again, I promise.
I already told them that for the reception, take out all the juices, take out all the sodas, just water.
He actually just kind of yelled at us about it, actually. So your questions, we'll start taking questions now for about 15 minutes or so. We've got 2 microphones down here at the front. I see one gentleman waiting impatiently to jump in. So please.
Very interesting talk, and I'd listen to another hour of it if you had more. My first question, I just have 2 if they fit the time, is in recent literature, it seems like some papers say that metformin may be a negative effect on cognition, and others say that metformin may be a positive effect on cognition. One, do you have any insight into that, and to what extent do genetics play a role there? And the second question is metformin for prediabetes. Is this patient-specific, and any thoughts on that?
Excellent. So for metformin and cognition, there's nothing really that proves any deleterious effects of metformin on cognition. Metformin has been around for about 40 years or so, and in the United States a little more recent. They approved it in Europe earlier, and we really don't have any anecdotal evidence of that being the case. Now, we need to study it, and so in the Diabetes Prevention Program, we now have the longest followed cohort. So the DPP began in 1987 or something like that. So we have a very long cohort of people treated with metformin for a long, long time. And so there are studies now being done in that cohort to see whether cognitive measures are any different. So far, everything seems to be negative, which is a good thing. Because it's a good drug. It works well. In terms of metformin's effects for diabetes prevention, all the genetics that we've done in the DPP show that there are some people who respond better and some people who respond worse. And in fact, if you happen to carry a variant in one of the genes that encodes a protein that gets rid of metformin, then you don't respond as well because you get rid of it too quickly. And so there is a number in our clinical practice. We see that about 20 to 30% of people fail metformin. And so wouldn't it be nice to know who they are ahead of time so that we don't waste our time trying a medication that may have some side effects and is not effective? And it would be great to identify those people ahead of time so that we can then change the algorithm, and metformin is not one size fits all.
So in the case that it doesn't work, the negative effects of metformin for prevention, are there any harms to any people with different genetic backgrounds of metformin?
So it's a fairly safe drug. So most people who develop side effects are kind of GI side effects. So you get a little bit of diarrhea, a little bit of cramping, and that typically gets better. So it's not really a bad thing. If you have bad renal function, it can be very toxic. But so we always check renal function before we prescribe metformin. But so far, there's not really terrible side effects. Some people also develop a mild form of vitamin B12 deficiency, which can lead to anemia. So that's also worth monitoring. Yeah.
Thank you very much.
You're welcome. Thank you.
Thank you for a wonderful talk.
Yes, thanks.
I have a question. Can you comment on the effect of gastric bypass surgery on diabetes?
Right.
And how it may affect it?
Right. So I lied to you a little bit. I said that we don't cure diabetes. And the one exception, we don't cure diabetes by medical means, but the one exception where diabetes can be cured is by dramatic loss of weight. And that dramatic loss of weight typically only happens when you do gastric bypass surgery, where you can have weight losses of 50 pounds or higher. And so we see that those people who may have gone to surgery because one of the indications is they have comorbidities or other diseases associated with their obesity, diabetes being one of them, get cured of their diabetes. And what's really interesting is that they get cured of their diabetes before they lose the weight.
Right.
And so there are pathways that are affected by the way the surgery is done, which basically rewires the way your gut processes food. So it takes a part of the stomach, kind of closes the stomach, brings the esophagus into the jejunum, so it bypasses into stomach, bypasses the duodenum, which is the first part of the small intestine, and then you kind of bring to them. So there's changes in the wiring and the signaling and the hormones that take place that all of a sudden cure your diabetes within a week or two. And so it is very interesting to figure out what is actually happening, and there's multiple active studies that are really trying to figure out what is changing, what's signaling. Maybe the microbiome, the bugs that we carry in our gut also change as a result of the rewiring. What is it that makes people be cured?
Is it the same effect with a Roux-en-Y versus a banding?
Only Roux-en-Y. Well, okay, the weight loss mediated benefit is from both, and it's purely dependent on weight loss. The immediate effect is only Roux-en-Y.
Thank you.
Yeah.
Hello.
Oh, sorry. She was making her way to the microphone, but okay.
Hi, great talk. Thank you.
Thank you.
My question is about epigenetics, how you think epigenetics plays into these different forms of diabetes, and if so, how epigenetics will play into the whole precision medicine field.
So that's a great question, difficult question. So epigenetics, for those of you who are not in science, is what genes are on and off. So we have the human genome, the same DNA sequence, 3 billion base pairs in every cell of our human body, including blood cells, and I can take that and measure it, and I know what DNA sequence you have, and that's the same sequence you will have for the rest of your life. Epigenetics is what genes are on or off at any specific developmental stage, whether you are a child or an adult, in what tissue of your body, in a different metabolic condition. So that requires measurement of such things in multiple different tissues. The ones that are relevant to diabetes are the liver, fat, muscle, brain, and beta cell. And then see what genes are on and off at different stages. So we've made very rapid advances in genetics because DNA was easy to collect and it's available in thousands and thousands of people worldwide, and they were stored ready for us to do these analyses. Epigenetics much more difficult, particularly in the tissues that we care about. And so there's no question, diabetes being a disease that genes interact with the environment, and the environment talks to genes via these epigenetic changes, that epigenetics will be very highly relevant. And where people have looked, they have found epigenetic changes that correlate with diabetes. But it's much more laborious and difficult to catalog all the epigenetic variation that is required. But that's definitely the next frontier of figuring out how does the environment talk to genes, and together they produce these changes.
Thank you.
Yeah. And we'll go here. Yes. Hi.
It was a great talk.
Thank
Thank you
you.
very much. This is really naive. Don't anybody laugh. Okay.
It's okay. No laughs.
No laughs.
Okay.
But I was amazed that it went back in history. I was that you had this Egyptian example and so on, and these populations where you would never expect it, as with the Inuit who don't eat sugar, as far as I know, or didn't. And so I wonder if the disease itself and these variations, do they represent an evolution of the disease? Is that a possibility?
Absolutely. Yeah, absolutely. So there's this thing called the thrifty gene hypothesis. And the way it's a hypothesis, but the way that works is as follows. So for most of human history, we've lived in times of famine. Right? So it was difficult to come up with food. You were basically hunting for the mammoth, and then the mammoth walks in, you hunt it, and you eat, you feast, but then the mammoth is dead, and then you kind of starve until the next mammoth,
And no sugar.
Right. And so what happens then is that as we evolve and we have to fight this very hostile environment, the traits that get selected are genes that help you store energy and be very efficient at putting energy in. Those are the genes that help people, and people who did that better reproduce more, and so that's how genetic change evolves in a species. Now, agriculture comes along, and then of course, caloric superabundance in the form of McDonald's comes along, and so now we have these populations that have been pre-selected to have genes that make you very efficient at storing energy, and so that now works against us. And so it's amazing how from about the 1960 or so we've seen this epidemic. And in some places it's radical. So in some of the Pacific islands that were taken over by the United States in 1945 after the end of the Second World War, like Samoa and Guam, in those places diabetes and obesity completely skyrocketed the moment we walked in. It's almost like it was an infection. And so there is definitely evolution playing a role in how obesity and diabetes have taken hold.
Thank you.
Yep.
Hello. I'd like to say thank you for coming out here tonight. Great presentation. I have two questions. My first question is, so you do a lot of work with genome-wide association studies. One of the well-known difficulties with these is that false positives are relatively common. This can be addressed by lowering your p-value threshold, but doing that you can oftentimes swamp out real but not so clinically significant effects. What do you do to address this? And my second question is, there's some evidence to suggest that patients that respond well to metformin that have diabetes tend to live longer than patients that do not have diabetes. Is there any mechanistic hypothesis as to why this is the case? Thank you.
So the first question is a more of a technical question. So for the non-genetic audience, when we look at the entire genome and look at variation that may be influencing a disease, we are performing, there's 3 billion base pairs in the human genome. So typically in one of these studies, we perform 1 million tests. So for us to be able to say that we really believe something or we don't believe something, we need to be really stringent, because the chances that you're going to have some of your genes come up to a certain level by chance alone, nothing to do with the disease, but simply because you've done 1 million different tests, are very high. So for us to say we believe these, we demand a very stringent threshold. So what the question is about is like, okay, you're probably making sure that you capture the wheat and get rid of the chaff, but there's probably a lot of wheat still left among the things that you have discarded. So how do you address that problem? And so the way you address that problem is by increasing your sample size so that you can get more and more things to come up. So we're doing that. You can also try to bring in other parts of biology that help you point among the things that you were able to discard. These things maybe you shouldn't discard because in mouse it has this effect, or because I know what tissue that is expressed that is relevant, and even though it is just below the threshold that I would have used to declare that I believe in it, I think I still believe in it if I bring in additional information that can make it come up. There's other ways by where you can do different ways of ascertaining genetic variation by sequencing as opposed to genotyping. So there's many different things that allow you to rescue some of the things that you would have discarded. But right now, with 100 different places of the genome for diabetes, we've only explained about 5 to 10% of the genetic contribution to the disease. So there's really about the models predict about 3,000 different parts of the genome that will contribute. Was that your first question? Yes. The second question is about metformin, and people who are treated with metformin living longer. So there's great interest in metformin's role in cancer. The target for metformin is not fully known, but it appears that it really does help with prevent cell proliferation in some ways. It interacts with the nuclear pore complex. And so there is a number of clinical trials in the US now, including the Diabetes Prevention Program, looking for cancer outcomes, which of course would have an effect on longevity. So it's possible that metformin may be repurposed for other uses if these trials become positive.
Thank you.
Yeah.
Thank you for a great talk. Throughout your talk, you mentioned that there's a wealth of data, both from external factors and the genome that we need to look at, and there are about 100 different genes that might correlate with diabetes. And I think the buzzword that comes to mind at this point is something about machine learning and trying to mine this data in a more automated way.
Yes.
So I was wondering if there are any initiatives that deal with that, or if you're involved with any of those kind of
so there's a big, something called Big Data Initiative by the NIH, which goes across many different diseases. There's a very large effort in diabetes. And the very first part of that is creating a place where at least all the genomic and trait information is stored in a single place. And so here at the Broad, we're constructing a type 2 diabetes portal, which you can Google, type 2 diabetes portal, www.type2diabetesgenetics.org. And it is co-funded by industry and by the NIH government, and it brings in pharma partners together with academic investigators and government scientists to really create this repository where this information is securely stored so it doesn't really leave the firewall, but then people from across the world can come and mine it and use some of these very machine learning approaches that you're suggesting. And we're going to bring in clinical information from some of the clinical databases that exist who are consented for this sort of thing. So it's all very compliant with all the ethical and privacy regulations. But the idea is to democratize genetics and make all of this information available to the world. And whether you are a high school student doing a work project, or a pharma scientist who tries to develop a new drug, or some clinician who needs to know what this mutation is causing the disease that he or she sees in the patient, all that be available to you. So,
I think we have time for one last question.
Hi.
So since diabetes is considered, or at least type 2 diabetes is considered to be a disease of insulin resistance, I'm wondering if limiting carbohydrate intake could affect outcomes for patients, or what your thoughts on that are.
Right, so it's not just insulin resistance. So let me just make the point. So yeah, so obesity gives you insulin resistance, but then you need to have a beta cell complement that is not effective. So if you have infinite beta cells, you will never become diabetic because you'll make all the insulin you need to overcome the resistance, right? So it turns out that, yes, you do have this insult of insulin resistance, but you need to have also some sort of limit, finite amount of insulin that you can make, maybe based on genetic predisposition. Okay. So it's a combination of both things. It's sort of beta cell insufficiency as opposed to deficiency combined with insulin resistance. But yes, of course, limiting carbohydrate intake brings glucose down. So if your measure is what your glucose is, then that's fine, but you still have the insulin resistance. And if the insulin resistance is what gives rise to the complications, heart or stroke or kidney, etc., then you haven't really done much by simply addressing the glucose. So you really we need to come up with medications that reverse the entire process that made you insulin resistant in the first place.
Okay. All right. Thank you.
Okay. So I think there's a reception, a healthy reception right outside. I recommend that you walk home. And I will be around if people have any questions. Thank you.
Thank you very much, Dr. Florez, and thank you all for your questions and for listening tonight. And as he was just saying, we have the healthy reception out here. And we hope you'll come back again on August 9th for the last talk of this year's Midsummer Nights Science series.